So we're going to start today with the Phage annotation. It would be great if there's an issue to just interrupt me. For the sake of the recording, your face will not be shown. It will only be the screen that is shared that is shown. So don't worry about that. And I'm gonna try and put all your faces all on one side. Okay. First recap from yesterday is actually we'll need to log into our server again. So we'll use SSH again. So SSH, I was Phage T1 because obviously I'm the most important person in the room. I will put in my password. Hopefully I'll have it right from the first time. Excellent. So I'm now into my program. Before we try to run any programs today, we'll need to remind ourselves we created a Conda environment yesterday called WS for workshop, where we installed all of our programs. So if we now don't activate our program, our Conda environment, we're not going to be able to run anything. So we'll do Conda activate WS. Now we can see at the front here that it's activated. All right, first step. Let's see in our instructions what we were going to do. So I had a little note here about orf calling, which is optional. And Andrew explained it actually really well, so I'm not going to go into details, but if you want to use the NCBI tool Orf Finder to just have a look at all the potential orfs that are in your phage genome, you can paste the first 50,000 sequences in here in the link, and then check whether how many orfs there are and how they overlap and whatnot. We'll not do that here. We'll immediately go to the step of assigning decoding sequences. So I'm a little bit of theory at the front. Andrew has covered that really well. So for the sake of this exercise, we'll use the software Proca. So Proca is kind of an automated tool that wraps a couple of different programs. The most important one thereof is Prodigal, which is the software that actually calls the start and stop sites, and it's a really good program for phages. It works actually surprisingly well, given how old it is, because it's one of the older programs out there. So the first thing that we do is, so we've pre-installed it as part of what you did yesterday, is just to type Proca to see whether it's installed and it runs. So I'm just going to type Proca. So what it shows now is the help file, and basically name of the program. It's written by Thorsten Siemann. It was originally made for bacterial genomes. And then there's a whole bunch of optional parameters that you can choose, but in the simplest form, you just run Proca and give it your context file. Now, one of the things that happened to me yesterday or the day before, as Andrea talked about, we had a bit of a problem with one of the databases, and we fixed it by running Proca setup db. So we'll just make sure that the underlying databases are set up. So I'm running that now. And one of the things here is that Proca actually uses UniProt as an underlying database. So it already makes comparisons with a very well curated database. But because it's such a well curated database, it actually will assign a lot of hypothetical proteins. Because it will do an automated step in which if it's not sure about the protein, it will follow Andrew's rule and just be hypothetical protein. So I have given here an example bit of code that we can run. And I'll run you through it now. And basically everywhere where I put the greater than signs is where you need to put in the sequence or the path to the file that you have for yourself, just in case you've given it another name and I don't know about it. So this is where it gets more complicated and we're not just going to copy paste.
Can I ask quickly, Evelyn?
Yes.
If you're looking at, for example, if you're looking at prophages, is it a good idea to go straight to the kingdom of viruses because there's far more data available in the bacterial genomes?
You can go to the bacterial kingdom. The thing is, so the kingdom that you choose, that will point it towards certain underlying databases. So for example, if you use kingdom viruses, it will have the capsid proteins and terminase proteins and those type of well-conserved phage proteins in its database. So if you use the bacterial database, I'm actually not sure whether they would be annotated or not. That's something that you can trial and error. It runs quite quickly, so you can just do both and compare.
Okay, thanks.
So we'll try this and see what it says. Okay, so what we're going to do is Proca kingdom viruses. One of these here, I'm choosing the genetic code 11, which is the bacterial code. So that's where we combine viruses and bacteria, meaning we have phages. Now, there are some phages that don't follow this code. There are some of the huge phages that have been metagenomically predicted and some of the crass phages. So if ever you have a phage and you use this and you almost have no proteins predicted, then this is one of the parameters to have a look at. Proca can add a locus tag, so that's good. And the rest of it is kind of just making sure that your files have a recognizable name. In contrast to what Andrea was saying for the previous set of programs, Proca does not want your output directory to exist already because it doesn't want to accidentally overwrite any files. So you have to make sure that the output directory does not exist. Let's get started. Proca -- kingdom viruses. Make sure that you write it with a capital, otherwise it will not be recognized. We're going to do G code. You can copy paste this bit, of course, but I'm just going to write it all out. Okay, locus tag. I'm going to do T4 today, so we're going to write T4 here. Obviously, that might already exist, so this is something to think about. I think still at EMBL and NCBI, the locus tags need to be registered at the moment you submit it. So it has to be sure that it doesn't exist yet. Prefix, will give a prefix to my file name. So I'm gonna also choose T4. I'm gonna make an output directory that is Proca _ T4. And then I just need to get the last thing is where my Fasta file is. And yesterday I put the Fasta file in the folder assemblies in my home directory. So I'm going to use the tilde to just refer to my home directory. It's where I'm at the moment, but just to be safe. Assembly, now I'm going to tap complete assembly and hopefully T4 Fasta is here. Excellent, T4 Fasta. And then I'm just going to let it run. And that's it, it ran. So now we can have a look at, I'm just going to clear my screen. I'm going to have a look at, is it the folder here? Yes, Proca T4 is here. So now I'm going to list what's in the folder, Proca T4. And these are all the types of files that are here. So we can have a quick look at what's in some of the files. We can open it with the less command, or we can have a preview of the first 10 lines with the head command. So I'm going to do head T4 dot. I'm going to change into the folder first, otherwise I'm not going to. So now I'm in the folder, you can see that because it's written right here. If any of you are on a Mac and are wondering how to get this nice blue terminal, it's called Settings Ocean. We were going to do head t4.faa to have a look. Okay, we can see, so this is the multi-Fasta of the proteins that are predicted, and we can see here that we have our locus tag is T4 underscore and then a number, and then our first predicted gene is an endonuclease. Interesting. So let's have a look at the GenBank file as well. Okay, you can see that it automatically inherits some of the information from the Fasta file we gave it. For example, it gives here, it says that it's just, that was the contig name. It gives you the total length, that it's DNA, that it's linear, which is important because our assembler said it was circular. And then, you know, it leaves a lot of it blank with defaults, and these things we can change later on. Is everybody still with me so far? No dissent. I hope we are still there. So then we have done this, looked at what's in the directory, had a look at the GenBank file. Okay, so I want to, before I do anything else, I really want to visualize my file now. So I'm going to download it, and I'm going to use CyberDuck. I hope I'm still logged in here. No, maybe not. Refresh. Okay. So I hope all of you who needed it saw the tutorial that Andrea made about how to use CyberDuck to download stuff. So I'm going to download the GenBank file. And I'm going to allow this, and it is now downloaded. Oh, now you can see, now you'll see all of the, oh no, you won't see this. I'm going to now, I'm doing stuff in the background. And I'm going to copy it to a directory that I'm going to use for Eugene. And I hope you all saw my note to download Eugene. This is Eugene. And I've made a project, Phage Annotation Workshop, which is already opened. And now I can open files. This is my Phage Annotation Workshop. So this is the t4.genbank that I just made. And if I open it, this is what I see. 3 different tracks, one track with all of the coding, well, all of the feature predictions, one track that is zoomed into the sequence and the reading frames and the translations, and then one where it's a summary of all of the features, and then we can see that Proca predicted 261 coding sequences and 10 tRNAs. We can use this here to investigate some. So we can, if we press on tRNAs, we can see the tRNA, and then we can see that Proca actually used Aragorn to predict these tRNAs, which is very good. I also like the program Aragorn. Okay, so now we're going to follow kind of the, let's say, the Kropinski method of scrolling through your genome, and have a quick scan at whether we think the coding sequence predictions are good. And I'm going to zoom in to something that is a manageable distance of about, let's say, 10 kb at a time. And we can see that it actually does look good. There are some gaps. Not too big. Okay, so for example, here, if you see a gap like this, which is not too big, and it's between 2 genes that are pointing outwards, that's not something to worry about, because this is a place where you probably would need to have some regulatory elements encoded. So you probably won't need to look to extend for an overlap. And actually, if you have an overlap here, that's a dangerous thing to see. I'm going to scroll a little bit further. And I'm not going to change anything now because the first thing that I want to check is whether the genome has been oriented correctly. Remember yesterday we did the phage term analysis, and then we saw that T4 circularly permuted, meaning that there's no defined end on which we can start it. So I've done a little literature search for you already, and we can find that here. So checking the orientation of your genome. For T4 is one of the most well-described phages out there. So there's a wealth of literature. And luckily I've worked with some similar phages during my PhD. So I did quite a lot of reading. So I remembered, okay, in this publication, the 2003 publication describing the genome, it says in the footnote of table 1 that the genes are listed sequentially as they appear in the GenBank file, and they start with the first base 5' of R2b. If you look in that paper and you look at the table, then you can see that the gene R2b is encoded on the complementary strand. So it's actually faced from right to left. So basically what we need to do is find the R2b gene, make sure that it's in the reverse position, find the start codon of that gene, find one base upstream from that gene, and reopen the genome at that position. So one of the things we can do is see with this function find qualifier, looking in this coding sequences whether in the value R2b is found in the annotation. And I've pre-checked this. Unfortunately, it was not found. And this is one of those issues with some of the annotations, because it's actually there. So let's look for the R2A gene, because that's right upstream from it. And the R2A gene is found. So the R2A gene is this one here, locus tag 48. And I know that this gene must then be the R2B gene. Now, in this case, Proca wasn't sure, and it made it into a hypothetical protein. But I've pre-checked this for you guys, and it is indeed the R2B gene. But while looking at it, I also realized that in the RefSeq sequence of phage T4, the product is no longer called R2B. Somebody changed it. And that's why, so this is where we kind of find this, when something is changed, snowball effects. So what we're going to do now is here, so I've clicked on this one here, and now you see it highlighted here, meaning that we were looking for one base 5' of the R2B gene, which would be this T. Let's try this T. Yeah, so now I've selected it. And I'm going to make a note of the position, which is 30,947. Okay, let's go back to the command line, because now I am going to use, I'm going to go into my assembly directory. Just so changing working directory. So whatever I'm running now is running in this assembly directory. And we have here T4 Fasta, T7, and T7 we restarted based on the phage term results. So now I'm also going to restart the T4 genome. From Eugene, we can see that we wanted R2B to be in the reverse direction. So we don't have to do a reverse complement. So what we've written here, the reversed complement is not necessary. So we can do the sec-foo rotate I, and then here we can use the 30,947 for the T4 Fasta and make it into a T4 restart Fasta, for example. So sec_foo rotate I 30,947 T4.Fasta. CF tab completed just to make sure that it's present. And we need the greater than sign. And then I'm going to do T4 restart.Fasta. Okay, that is made. So now, phage annotation is a highly iterative process where each time you find something, you might need to redo something. We're going to have to rerun Proca. And as Andrea showed yesterday, you can use the up arrow files to see some of the commands you've done. Oh, Carlos asked if I could quickly remind why I rotated at that location. Okay, so this is, we are now reorienting the phage genome based on the closest relative, which is actually kind of itself, because this is a version of T4 that we've sequenced that was in our lab. So if we then go check literature for T4, so let's say you have a genome that's not T4, but quite related to T4, then you go look at the publications. You can also go look at the GenBank file of T4, and I have it, and I'll also open it now. Open, I have, this is my reference T4 GenBank file. And we can go look for what the first gene is, and that's the R2A gene. So we want our genome to be oriented exactly the same as a well annotated genome in the reference database. And because we know we're sequencing T4 and T4 is already in there, we can look at doing it that way. And because I know that for T4, it's well described in literature exactly what the decision-making process was of opening it, I know that, as I described here, it's the first base 5' of the R2B gene. So that makes it very easy to locate, because I have here, so this is my R2A gene. This will be the new first gene. This is the R2B gene. This will now be the last gene, and then which is oriented from right to left. So it's on the bottom strand. It's, if we select it, you can see that it's here. So 5' of something that is oriented from right to left is on the right side of it, which is this one. If I select it, I can see that it's in location 30,947. And that's how I decided to do the sec_foo rotate with 30,947.
So if the first person chose a stupid place to put the start, like in between two genes which are clearly related to each other, everybody else copies them?
Yes. Yes, exactly, Melinda.
Oh, we could just start with the terminates.
Yeah.
Question, because I thought that when you use phage term, it also gives you the right orientation of the phage genome.
That is only for genomes that have, for example, a defined end, or which have a defined packaging site, which starts packaging, and then you can find the orientation. In the case of T4, it is circularly permuted, and it has no kind of preference for direction of packaging or direction of any type of transcription. It is also one of the first phages found and described. So anything that happened for T4 is sometimes not intuitive, because it was analyzed before we had any genomics. And in this case, we'll just have to suck it up and follow what people a couple of decades ago did. So, for example, some of the Ackermann-Viridae phages, they also have R2A and R2B. And there it was decided to open it upstream of the R2A gene so that the R2A and R2B are together, and have them be listed from left to right. So it's a completely different orientation. And that was, sometimes it's good to be first, because then you can do your thing and everybody just has to follow you. And this is just good practice to make things in a database more consistent. And I would very much urge you to do this because of that good practice, not because somebody will reject your paper. Although if it's Andrew or me, we might reject your paper or ask you to do revisions. Okay, is everybody with me on the rotating and restarting the genome? I think that's a yes. So what did we do? And Andrew makes a good point that the consistent ends are important because it helps with comparative genomic analysis, and it does make your plots look a lot prettier as well. Okay, so we have now done the seq-through rotate. So we have a new genome called T4 restarted Fasta. So now we need to run Proca again. I'm going to use the up arrows to just go to my previous Proca run. And I'm going to change some names. I'm going to make the output directory Proca restart. And I'm also going to use Proca restart.fasta. And I'm going to leave everything else the same. And it looks like I didn't make any typos. So now we have a Proca T4 and a Proca T4 restart. Just quickly checking what's in the Proca T4 restart. We have all of our files created, so everything should have run fine. Now, because I didn't change my name, I now have two t4.genbank files, which, if I didn't copy them into a different directory, might cause some confusion, but that's just me. Maybe I should have changed my file prefix. I'm now going to, but let's do something else. Let's just replace this original file with the new one that I'm going to download. And I'm going to use CyberDuck again. So I'm going to refresh. So Proca restart. And I'm going to just overwrite the T4 GenBank, and I'm going to replace it into my folder, and I'm going to overwrite. And then Eugene will tell me that my T4 was, do you want to reload it? Yes. Okay, let's see Eugene. So this is now the new one. First gene, what is it? R2A, excellent. We are now in the correct orientation. So now we can have a look at our coding sequences. And I'm going to do an iterative process to just show you how often you kind of find something out and then be like, oh yeah, but maybe I should run this again because now I have more information. Because one of the things that I wrote in the guidance is to not yet run Proca again, because we know we need to run Proca again, but there's a way to improve your functional annotations. So we might as well get started on that again before we get too busy with our Eugenes. Because if we go back to the our files that we have run, I'm going to change directory into the Proca restart directory. I'm going to do less long so I can see them cleanly. And I always like to have a look at the t4.tsv file because it quickly shows you all of the features that are predicted and whether they're all hypothetical proteins or not. So I'm going to use the less command, t4.tsv. Okay, and then I'm going to see we have the correct protein at the front. We can see that the CDS product is called protein R2A, and the gene that corresponds to it is R2A. So we can see lots of hypothetical proteins, some predicted proteins, some hypothetical proteins, but there's a lot of hypothetical proteins. So it might be a good thing to add a different database to Proca to improve our annotations. So one of the things that comes pre-packaged with Proca is that you can use any type of well-annotated reference genome or protein catalog to update the functional annotations with the hope that there are fewer hypothetical proteins in your end result. In this case, what we will use is the T4 GenBank file from NCBI. And what you can do is you can use the accession number for the original T4 and look into NCBI and then download it and upload it into your VM. I have it here somewhere in one of my own folders. I'm just going to have to rename it to T4 reference. And I'm going to, you probably won't, yes, sorry, no. When you, in the background, change files for in Eugene, then it gives you all of these type of, you know, oh, if you've changed something, do we need to change as well? So I'm doing that now, T4 reference. So I'm not going to save anything. So we're back in CyberDuck, and I'm going to, I'm in my home directory. You can actually also use CyberDuck normally to make a new folder. I'm right-clicking, make new folder, and I'm going to create a new folder, reference, so which is currently empty. Yeah, it's empty. And in this folder, I'm going to drag and drop my T4 reference genome. Okay, so sorry, I keep getting these pop-ups that you probably won't see. Okay, so now I have in my home directory a folder called reference. If I go to my home directory, I'm going to list. So now there's a folder called reference, and in this folder reference, I have the T4 reference genome. So what I can now do is use these annotations to run Proca again and update my own genome annotations. So again, we will use Proca, kingdom viruses. I've now, to make the difference between, you know, T4, the actual T4 as is in the database, I decided to make it now T4 QIB with the prefix T4 QIB, because later on you will see that this T4 has been passed around labs so many times that there's quite a couple of differences between the original T4 that's in the database. And what we need to do is use the proteins flag to tell Proca that what the GenBank file that we've used is what we want to use as a reference. So don't copy this one literally, because it will all depend on what names you give it. I'm also not going to copy it literally because I've changed some of the names, but I'm going to copy this bit already. So then that says same. So Proca kingdom viruses. Oh, I forgot the genetic code 11. I'll add that in a bit. So proteins, it was in the folder references and it was called t4 reference.gb. I forgot to add my genetic code, so I'm quickly going to genetic code 11. It doesn't matter in which order you do this. I'm going to have to specify a new output directory because Proca doesn't want it to already exist. So we can call it Proca, we can call it better Proca T4, for example. It's all about, I always like to give my stuff names that I can later on remember what I was doing. And then we need to give it our Fasta, which was in assembly, and it was called, so it was in directory assembly, and it was T4 underscore restart.fasta. I think we have all of the stuff we need, and let's just rerun it. Okay, if we scroll up, one of the things that I wanted to point out to you is that Proca does a lot of cleanup already. So here you can see in the log what all it has cleaned up. So for example, where a product was called GP 5.1, conserved hypothetical protein, Proca automatically changed the name to hypothetical protein. So there's a whole list of things that Proca already cleaned. For example, conserved hypothetical predicted membrane protein, hypothetical protein. And so thank you, Proca, for already doing lots of the stuff that we would otherwise have to do by hand. Okay, let's now check CD better Proca list. We have all of our files, and let's check the table file again. Less T4QIB TSV was the, yeah. Okay. Oh, now we can already see that there are a lot more things that were predicted. Not necessarily in a better way, because let's have a look here. So we have the MOT-B gene that is now found, which is good. But it also found the two genes downstream from it, MOT-B 0.1 and 0.2. And this was just done in T4 to say, okay, we found some hypothetical proteins and they were, you know, close to MOT-B, they were downstream from MOT-B or upstream from MOT-B. I'm not sure now. So in this case, Proca called this the MOT-B 0.1 hypothetical protein. It does the same for the two proteins upstream or downstream from DEX-A. It does the same for, you know, the proteins upstream, downstream for MOT-A. So while we do have some additional predictions, that doesn't always mean that they are correct or informative. In this case, we know that we just have the same protein present as in T4, and it was not greatly annotated in T4 either. And here we see, because we are dealing with T4, and in T4, there's a lot of proteins that have been annotated with the GP function, as Andrew said. So our suggestion would be to move this information about GP to the notes section. So we still have quite some hypothetical proteins, but they're probably also hypothetical protein in T4. And the first time people see a phage genome, people are usually afraid of all the hypothetical proteins, but that's entirely normal because phage proteins are just not as well characterized in their entirety. Okay, excellent. We can find quite a couple of the structural proteins as well. base plate wedges, subunits, and the neck and the terminase. Excellent. The terminase small subunit, the large subunit, which is all good. I'm going to press Q to quit this. So let's now copy the T4 QIB to my file, to my home system, so that I can better Proca. I'm going to choose the GenBank file, and I'm going to drag it into my folder that I'm using to run Eugene from.
Sorry,
Okay.
Evelyn, can I ask a question?
Yes.
So here, obviously, you're annotating it on the basis of itself, so you know it's correct. But if you've got what you think is a related phage, you don't know how related it is, do you have to include any percentages or cutoffs or anything to say only annotate this protein if it's of a certain level of homology?
It's a very good question. So I'm using this as an example of how to improve Proca. One of the things that you can do is there are catalogs. You can, for example, if you don't know what protein, what phage you have, is you can give it all of the isolated phage genomes, for example, in a single database, the whole phage catalog. Or
aren't you doing that already with using Kingdom viruses?
No. So that's a thing for Proca, is that the standalone vanilla version of Proca, as it gets packaged, it comes with a very, very limited database that it compares to. And that's why a lot of the proteins are hypothetical proteins. So the way you would improve the automated proportion of your Proca annotations is by adding as much information in databases as you can, to the point that you can give it a gigantic database, but then it will probably take a long time to run, and it might be too much for your system. So it's kind of a balancing act. Now there's built-in evalues that you should be able to change. So if we were to go to the Proca manual, and I'm gonna check whether there's I'm just gonna have an e-value. Okay, so it has a setting where you can change the e-value. So if you want to change the setting of the e-value to 10 to the minus 5, then you can make a decision on, okay, if it's higher than that e-value, then it won't be reported, for example.
Okay, thank you.
So one of the things, but that's already in the wizard settings, for example. But it's always good to have a look at help files, and particularly all the tools by Thorsten Seemann are very well. The manuals are very good. Okay, so we didn't do this because we know that we're working with T4 and we're kind of comparing it to itself, as you said, Melinda. So I'm going to have a look again. And this was the one that I did now, T4 QIB, open. Okay, just to double check, my first gene was still the R2A. Okay, now I'm finally gonna have a look at the start side. I'm gonna zoom in again, oh, maybe not that much, to a manageable zoom depth of 8 KB. You can see here that this is the depth. You can use this, I don't know if you guys see this, but there's this tiny gray line here at the top. This shows you the depth of your annotations across the whole from 0 to 165 KB, meaning that if there's gray, that means that there's a gene predicted. Let's just zoom out all the way, and then you can see it in the same scale. So if it's all gray, that means that there's at least one gene or feature predicted. That can be a tRNA, a coding sequence, it can be a promoter or terminator. We haven't added those, so they won't be there. If there's a blank spot like here, that means that there's nothing there. So annotation density is 0. So we can, so I've now clicked on it. Here are my tRNAs. I'm going to have a look. If I double click on this, then the gene is selected in my track here. So then we can look for stuff that's going on upstream. So I can immediately see that extending this coding sequence, which is facing, sorry, let's just zoom in a bit, which is facing to the left-hand side. So the start is on the right here. We cannot extend it because there's a stop codon here. So there's no way that I can extend. So that's not a thing that I need to look for. There's also a good ribosome binding site predicted, so I don't have to worry about this. So then it's the matter of looking in this empty section whether there is a longer frame that could be something. Now, there's a way to predict ORFs in Eugene, but it usually does it for everywhere all at once, and it's kind of sometimes confusing and makes your computer go a bit slow. So I'm just going to look for long stretches in all of the 6 frames between stop codons, and the stop codons are the red dots. So I'm going to scroll here, and I'm going to see. Oh, here's quite a stretch, no, isn't it? Oh, interesting. We'll keep on going. We still, here in the last frame, we still haven't found the stop codon. Still haven't found the stop. Okay, here there's one. So basically this section, please scroll, yeah. Oh, up until here. Oh, I started at the wrong start type. Is an ORF. I need to start exactly at the initiation codon. So we're gonna use this one for now. So I'm gonna have to make sure that, yes, now I've selected the correct frame. And up until here. And what we can do is now we're going to select it. I'm gonna look, well, this is it's GGAA. It's not an ideal ribosome binding site, but it's still decent enough, in my opinion, to be interesting, more interesting than having an empty space here. So what I'm gonna do is I'm going to right click on this, and I'm going to add a new annotation. And it's going to be a coding sequence. And I'm going to not give it any description, and it's on the complementary strand, and I'm just going to save it to the existing document, and I'm going to create it. Voila, now we've added an additional coding sequence. But unfortunately, I haven't figured this out. If anybody does, let me know. I haven't figured out how it automatically incorporates the translation. So what you need to do now is you need to copy paste the annotation amino acids, and then add qualifier. And then this is the translation. And we give it what we copy pasted as value. To meet the start codon, we're going to change this to methionine. Okay, and now we can see that at least that much is now in our annotations. if you compare it to what's in the normal ones, then you have, codon start, inference, locus tag, product stuff. So what we might want to just do as well is we add another feature, another qualifier, sorry, add qualifier, and we will add a product that says hypothetical protein. Okay. And we can see that it's between locus tags 127 and between 127 and 138. Oh yeah, it's because the tRNAs also have their own locus tags. So we can call it something like 127.1 or something for now as a placeholder. Sorry, I'm going to make this slightly bigger so that we can see. Are you all still with me on what I'm doing and why I'm doing it? Value T4 QIB 100. What was that? 27.1. Okay, just for now. Andrew said that we can select a specific region in Eugene and look for ORs. We'll do that as an example. Sometimes it really doesn't work for me, but we'll just choose this bit at the top where we just predicted it and do where was it? Oh, I think it was here, right? Look for ORs. No. Look for ORs. Yes. Strand both, minimum length 75. Max results. Let's just do 20 or something. 11 region, whole sequence, selected region, and see whether it works. I guess somehow this always fails for me. And that might just be my version not wanting to listen. Let's have a look whether they were predicted and they're just not showing. Aha, morphs. Oh, for some reason, they're not showing up on my top track. So you can see here that it, you can see like Andrew showed in the codon stuff that he had in his presentation, that you can see the coordinates here. So all of the ones that are on the complement, there's a couple of small ones because you can see the coordinates here. But I think we found our potential gene here because here we're moving on to tRNAs. So I don't think it will be, there will be another one here. I think we found our extra one. We have some questions in the chat. Do you have an automatic way of searching for ribosome binding sites? So one of the things that we can do is we can use the basically a recognition sequence and do a sequence search. Because it's so short, it's not always accurate. And it sometimes finds too many things, but I have given some information about ribosome binding sites. Where have I given it? Let me look. I know it's somewhere. Yes, no, shouldn't. Yeah, okay. So there was a recent paper by Krishnamurthy and Wang where they checked all viruses for ribosome binding sites, and they found that near 100% of the bacteriophages had this sequence AGG, or a 4-mer or 5-mer permutation of this sequence in front of their morphs. So if you look for either the whole sequence or the GAGG or GGAG, something like that, then it's a good chance that's a ribosome binding site. Back when I started, we always just looked for an AG-rich sequence upstream, just to make sure that we didn't miss anything that you miss with automated tools. Tegan asks, so the coding sequence can start from a codon which is not recognized by translation table 11. In this case, it is recognized. ATA is part of translation table 11. It's just that the predictions from the automated tools vastly prefer ATG and then and then GTG, TTG. But it is part of the translation table. Okay, where have we got? So basically what you need to do, what we're not now going to do, is scroll through your whole genome. But I've had a preliminary look, and it's actually very good compared to the actual T4. So there's very few regions where there are any problems or where we have to look to extend it. But I hope that Andrew's lecture and my scrolling through here kind of gave you an idea of where, what you need to look out for. Because of course this is T4, and T4 infects E. coli, and both T4 itself and E. coli are some of the most well-studied organisms in the world. So all of the tools will obviously be better for this organism than for some of the other phages. Melinda, I don't know, because I've never used Artemis, to be honest. So if somebody who
okay,
is...
so they have a button where you can extend the start site upstream to the next start site, given any codon.
Okay.
And can trim it to the next any start site.
Okay, I don't. So I don't.
It's just really quick.
Think this has, because you can edit the annotation, but you need to know the coordinates that you want to edit it to, or you need to do that by dragging and selecting.
Okay. It's just that if you have that, then you can very, very quickly check the ribosome binding sites of all of the start codons in the gene, up and downstream.
Okay. In this case,
use Artemis.
Well, I've never actually used it because it was always throwing up errors on my Mac.
Get used to it.
So, but there's no one, and I always repeat that there's no one single good tool to do stuff. Although I do enjoy using Proca. But as you may notice, I don't do this often myself anymore. So I always have to struggle a bit. Let's look for one that we may want to extend. But again, the annotations are really good. Okay, maybe this one. But so we actually have an excellent kind of ribosome binding site. So I wouldn't want to move here. So I'm not going to do this because this bit is really good for me. Which the GAGG is better to me than the AGGG in front of this one. So I'm going to leave it. And a lot of this is kind of, some of this is down to kind of people's own personal feelings. And the only way to legitimately say what the correct answer is to do RNA-seq. So maybe, you know, the debate for this versus this is not worth it in this case because it's such a small extension. Okay, let's move on. So we've now done Proca a couple of times and we've looked at our data. We've added the additional functional information that we've added came from the original T4, but you can, as Andrew said, use multiple programs to manually add other information, other functional information to your product names. From NCBI, you can use your BLAST P or your SciBLAST. It also includes, you know, looking at the conserved domains database. You can use HHBRED or HHBLITZ. There's a new tool called Step 3 from Monash University, which is specifically developed to identify which are the virion structural proteins, which might be interesting for any of you. And then there's programs like Andrew said, like Phobias or TMHMMs to look for transmembrane domains, signal P. I suggest that you just try it a couple of times with a couple of your proteins just to see what it does. And if you have your own phage genome, then do it for all. You can make changes to your GenBank file directly within Eugene by using the add and edit buttons to add new features and qualifiers and change names. So now we've done that, I want to have a quick word about the regulatory elements. So I first want to have a look at the terminators. So what I'm going to do is I'm going to open the program Arnold, and I'm going to run my Fasta file, which I still need to cyberduck download, which was in assembly, T7 restart. Oh, T4 restart, sorry. And it is downloaded, and I'm now going to upload it. T4 restart Fasta. And okay, so this now is looking for those specific row-independent terminators that have the stem loop structure and the poly-T or the poly-U-tail on the mRNA. So we know that in our case, we're looking at T4. T4 encodes proteins on different strands. So we will search both strands. If you know that you have a phage like T7, which encodes all the proteins on one strand, you could choose to only search the forward strand. We're going to color it, we're going to keep it, and we're just going to run. And then in the meantime, I will look at a question. Annotation tool to is there any possibility to apply this annotation tool to prophages without inducing data? So one of the people that is in this meeting who is trying to do a lot of these things with prophages is James. So one of the things that he does is use a program called Vibrant to identify the prophages, and then if you have your prophage sequence excised artificially from your bacterial genome, you could just do whatever we're doing right now with the prophage genome. The thing is that you don't know is whether the ends of where it was in silico excised are correctly predicted by the program. If that makes sense. Hani asks, are we leaving the ones that were called mod B and mod B1 and mod B2? No, we won't. We'll eventually have to go through the whole file list and change a whole list of proteins and change what we don't like. And that might be, because if all of them are called differently, that might be a relatively tedious task. It can be done in Eugene. It can be done in WordPad, Notepad, text edit, whatever you would like. So in this case, here, this coding sequence, I don't like, what do I, oh no, it's hypothetical. So it's good. The product is a hypothetical protein, but it just has a note to the GB 4.5. Oh no, actually, maybe all of them are correct and all of the information is in the notes. That's something to check. It might be. Let's do the search for the find qualifier, and we were looking for, was it mod B? Wait, what was it, 0.1 or 1? Just do mod B.
It was 0.1 and 0.2.
Okay, 0.1. Let's find qualifier. Mod B 0.1 next, so it's this one. Why? Okay, it's not this one. Let's just not select anything. Not select anything. I'm just looking all of the features. Find qualifier. Mod 0.1. Sorry, I'm struggling.
If you look on your TSV file, then you can find the position relative to the start.
Yeah, let's just look at that. Because the search function is not always great, in my opinion. Less T4. Oh yeah, it's right here. Okay. So it was number 11 and number 12. So that's number 11, which is here. So yeah, mod B.1 hypothetical protein. So yes, we'll have to change this, and we will edit, sorry, edit. Sorry if this is out of your thing. So edit product hypothetical protein. Okay. So yeah, because everything is, you know, different, has a different name, you can't just do a string search and replace everything. You might want to, now that I've deleted it, add a note saying, okay, this was similar to, you know, mod B.1. In this case, Proca added an inference line saying it was similar to the amino acid sequence T4 reference GenBank, and then it gives you the protein accession number. So this, well, it's not necessarily the greatest description. I find it very informative, so I might want to leave it. This inference line is also similar as a note line, in the sense that it's just a text string and it is not used for anything else later on. Okay, meanwhile, Arnold has finished running. So the output files look like this, as Andrew has also shown earlier, and it uses two different underlying programs to look for hairpins, RNA motif, and ERPIN. So when you have a lot of them predicted, one of the things you can do is you can already say, okay, I don't want anything, I want to be very stringent at the moment, so I'm only gonna look at those that are predicted by both programs. I'm only going to look at those with a free energy below -10 kilocalories per mole. And I'm only gonna look for those that have a poly theta, for example, just to begin with. So in this case, this one would be a good one to have a look at first. So I'm just going to select this and copy paste into Eugene. I'm going to use the search function. And it automatically highlights it, and I can see that it is in an intergenic region, and it is downstream from this gene. And to be honest, for me, that looks like a pretty decent location. So I'm just immediately going to add this as a new annotation. And in this case, there is one for terminator. So it's no longer coding sequence. I'm going to make a group name terminator, for example, and it was on the complementary strand exactly, and I'm going to create this. So now you can see it's in a different color, and it comes when you look at my features that were predicted here. I'm going to close. So now I have a new group called terminator with a terminator here. You can again, and the only annotation now is the location. You can add additional qualifiers by pressing add qualifier, and then you could say a note predicted with null, for example. This is basically up to you, but I wouldn't with these ones not necessarily add too much information. You could say that it's a row independent terminator, stem loop. So idea is that you kind of go through the whole document to add these little features where they lie. Let's do one more just as an example. Maybe I'll take a crappy one to see what a crappy one looks like. We're gonna take one with a high free energy, meaning that the stem loop's not as stable, and one that it has no poly T tail, or not a great poly T tail. Where are you? Maybe just check this one. So now this one's in the middle of a gene. So I'm just going to completely ignore this. And this is, there's 86. So this is why I already use my own internal criteria to see which ones are more likely or not. Then the final thing, the final feature that we're going to search for today is the promoters. And that can be quite tricky, as Andrew said. And what we're basically looking for, if we take into account that it can be host promoters or phage promoters. Now, T4 doesn't encode an RNA polymerase, so it will not be a phage RNA polymerase specific promoter. So you can use, pattern recognition, or you can search for conserved patterns upstream from your coding sequences. Now, Andrea has written a new easier program to get these upstream sequences from the GenBank file, and it is already installed on your VM. So I gave an example here, but obviously I've changed quite a lot of names. So we're going to look for the let's make this a little bit shorter and clearer. Okay, so these were our files. We were going to look in the GenBank file, so we're going to do get upstream, and it's actually installed. Input file I is t4qib.gbk. We're going to name our output file upstream T4, and we're going to look for the 100 bases upstream. And it ran. So in our list, we should now have upstream T4, with which, if we have a look at the first 10 lines, looks like this. Okay, so it each time gives the name of gives the locus tag, it gives the coordinates, it gives the strand, the product if there is one, 100 upstream, and Andrea may want to say what the slice means, because I'm not quite sure what that means. And then so it's a list of 100 base pair sequences that are all upstream from a coding sequence. I'm going to have a look at the tail as well, because it might be that the last one we'll have to maybe manually cut, because there are no sequences anymore here. Okay. Let's download this, because I want to use the meme suite, as said by Andrew. Where's my CyberDuck?
So to clarify the chords versus slice, chords is the original gene location.
Okay.
While slice is the upstream bit coordinates.
Okay. Okay. I now have the upstream. I'm quickly having a look at it, whether it downloaded while on my computer, but suddenly my computer is being a bit slow. Okay. I'm just manually, you can't see it probably because I'm not sharing that, but I'm manually deleting the last one in the text edit file, because I know that it will give a problem when I load it into what we're going to use is the meme suite. So meme stands for multiple motive discovery elucidation. So we're an elicitation apparently. So we're just going to run it in default mode. I'm also very much a fan of default modes. Choosing my file upstream. I want to look for 0 or 1 occurrence per sequence. Meme should find me 10 motifs. And I'm not even going to bother with the advanced options at the moment. And I'm going to start my search because I just want to show you what happens. In the meantime, have a look at the chat. Melinda asks, why is the terminator definition valuable? It is a good question. It is, it's basically, it will actually allow you to refine your coding sequence predictions and start sites as well, because you will, once you have predicted all the terminators, you can know when you have a terminator here, then there shouldn't be, your coding sequence shouldn't run necessarily into that terminator, for example. In terms of the biology, but the most important thing would be in terms of the biology, I would say, where you can, for example, define operons of genes together, or in the sense for the T7 promoter, as T7 promoter is one of the, I'm guessing one of the most used promoters for molecular biology. And if nobody had searched for it and found it, then we wouldn't use it. So there's a lot of you know, interesting things to do with promoters and terminators. A lot of phage morphs overlap indeed, and that's why you usually will only find the terminator at the end of a sequence of overlapping morphs. And Andrew suggested that genome 2D includes terminator predictions as well. Can you still see my screen? Because,
yes, we can see the browser.
Okay, because I've lost the green band around it that shows what is showing, so for some reason. So let's have a look whether, okay, so we don't have our results yet. Sorry. We're still parsing. Anybody has any other questions? Is everybody able to run stuff alongside me, or are you just watching what I'm doing? Watching. So now we're waiting. Oh, maybe I should have chosen T7 for this, which is shorter. So, what am I going to do here? Oh, you can also, one of the features that I haven't shown you here is that you can have a circular visualization of your genome. So it's a quick, that's also a quick way to look at it. But unfortunately, it will always give you the locus tags like this. So it's a bit of a crowded figure, but if you want to have a quick circular figure, this works really well. One of the other things you can do is if you don't like the colors, you can change them. So now the coding sequences are in purple, but if I were to not like purple, but I do like it, so I'm going to keep it, then you can change it. Terminators are now in yellow, but maybe I want them to be in more purple. Yes, this is going to be a magenta. This is going to be a theme. So these are now here. If I want to hide my circular view again, I'm going to just drag this one up and gone. Okay, where are we with the meme? Oh, it's a heavy server day, I think. One of the things that I haven't shown you yet, which might be interesting, is that there's also a built-in dot plot feature here. So you can build a dot plot you can choose which ones on the x-axis. So I have only loaded one sequence. So obviously, let's say I want to compare it, and this will be interesting for you guys, to the original T4 reference genome. And you can, I'm just going to leave everything as a default. And then I can see, haha, interesting. So this is my dot plot. We can see that the start and the start for both of them are exactly at the same spot, because they both start here. But then our genome is actually quite different at the nucleotide level from the original T4 genome. And that's probably because this has been a T4 that's been passed through labs for decades. So for the future, for Tegan, this would be an interesting side project again, but I shouldn't say this. So you can see that there's quite a lot of gaps on the diagonal. If everything's 100% identical, then you just have one black line. So there's a stretch.
This is the case for T7.
Is it also the case for T7?
No, T7 is a straight line. It's perfect.
Okay.
But I checked on IGV the differences, and they are all sort of mobile elements or something that makes some sense to be jumping out the genome.
So one of the things that T4 is kind of known for is that it has kind of hyperplastic, or T4-like phages have regions that are more prone to be mutated or different between different phages than because it's a very large genome, and as you have seen, it's not in this very rigid setup of a whole operon of early genes, middle gene, and late genes is kind of a bit more splashed out throughout the genome. So potentially those bits where there's differences might be because of in those regions with mostly hypothetical proteins, for example. Okay, are we done here? No. Of course, when I tried this beforehand, it was like done in 2 minutes. So I will try to entertain you in the meantime. Does anybody have, so you know, the whole process of doing this annotation well and to high standards is a process that can take a couple of days of, you know, dedicated work. Obviously, I can't do that now because you'll have to go through all of the genes and, for example, and, you know, but I hope we've given you now the background and the resources to do it yourself. And Anna is asking, can you please repeat how you got into the straight line? Yes, so this is a feature of Eugene called build a dot plot, which is this symbol here, or you can get to it from the file bar, but that's, you know, in Mac and Windows, that's a different location. So I'll just click this one, build dot plot, and then you just give it what you want on the x-axis and what you wanted on the y-axis. So on the x-axis, I gave it my own quantig genome, and on the y-axis, I uploaded the GenBank file from T4. If we look for smaller bits, maybe we find more identity. And if we don't look for 100% identity, but look for 98% identity, let's have a look what happens. Okay, it looks like it got, well, okay. Well, that's better, isn't it? So because we were looking for in our original diagonal dot plot, we were only looking for stretches of 100 base pairs that were 100% identical. Now we were looking for stretches of 50 bases that were 98% identical. So we are allowing for some point mutations, and now you can see that there's more of the diagonal that's filled, which is kind of interesting. Aha. And while we have done that, our mean has run. So I am going to look at the HML output. And this basically gives you, we've asked for 10 motifs that we were looking for. And the first one that we find is actually the promoter that Andrew used as an example, because it has the GTTTAC minus 35 box and the TATTAT box, the minus 10 box. Because we've clipped upstream, our start codon is actually here, it's fallen off. So there's a couple of, I'm going to see if I can, oh, there's not that much more information here. So there's 4 motifs with a good e-value. So this one is predicted at 36 locations, this one at 20, this one at 52. And if you have a look at this, this is actually our ribosome binding sites. And then there's this minor thing here, which I'm not quite sure what we're looking at the moment yet. And then there's a couple of motifs with, you know, e-values that are too high to now be considered, and only in a couple of locations. So let's just ignore it. So with this one, we were able to predict just based on agnostic pattern searches, our promoter sequence in 36 sites. And then we can have a look at, you know, visual representation of all of our, you can see our locus tags here. and we can see the motifs that were present in color codes matching to these up top here. So I think this red one is this promoter sequence that we've identified. So you can see, you know, the locations where they are. And the green one is our ribosome binding site. Does anybody understand everybody understand what we did here? So if we want to, we can, we should be able to download this information in some sort of text file as well. Andrew said, if you know which genes are likely to be middle or late, you can selectively look at the meme output. Yes, exactly. So there's a lot of, you know, T4 has a lot of information that's already known for it. So it has different types of promoters that are early, middle, or late. It has a set of genes, you know, it's well characterized. So we actually know already all of the locations and all of the a lot of the promoters. This is just a way to do it now we did it in a pattern agnostic way. Hani asked how to apply the two mismatch rule. In that case, you would have to look at the locations of these 36 sites and verify what the actual sequence is in that location. So in this case, when we click on it, see if it, oh no, it doesn't stay. We can see that this one specifically has GTTTAC, so that -35 box is identical. And oh no, this is just a motif, sorry. Let's go to Eugene and look for this one. T4QV002. Eugene, close. I'm not gonna save. Okay. What are you doing here? Okay. Zoom in at the front a bit. Not that much. Oh, are we looking at the correct? No, we're looking at straight at T4 now. I'm going to load the sequence again. Okay. It was 2. So we were looking in this area. So it's basically here that we're looking for, right? Shall we do a text search? GTTT, okay. GTTT, AC, and see if we can find it directly there. Okay, so it is right here. GTTT on the reverse strand, and then the other one is right here. So in this case, this method, by just using mean, will be very time consuming to do the two mismatch rule, because you basically have the motif that's discovered. In the motif, it has some room for error, and I'm not quite sure what the built-in mean rule for error is. So that would be something to go check in the settings. We can have a look at the settings again if we open it again, because we ignored the advanced settings, advanced options, because oh no, you can't choose the mismatches. No. Okay, so if you use this method, it will be difficult to do the to nucleotide mismatch rule. If you do a pattern search, because you can also use pattern searches or your very short blast searches, but then your evalues will be really high against yourself, then you might be able to use that rule better. And Andrew has given a lot of alternative resources to look for promoters, so they might be able to incorporate it. Okay, so to recap, we have looked at the coding sequences, we've reoriented our genome, we've run Proca again, we've then run Proca again, and we've looked at visualizing everything with Eugene, checking our coding sequence annotations. We have not done the checking for additional functional information because it will you can spend as much time on it as you want. There's a lot of good internet resources for that. But I have shown you how you can change in Eugene the information that you find with these additional functional searches. We've touched on the terminators and the promoters, and I've shown you a way to add them. So if we go back here, it was the GTZ CAC, so you can add new annotation. So it would be your -35 signal that you would add here as a complement. So that's a new group that is now created of types of annotations. I want it to be in orange, and now it's there, for example. So now that we have done that, so the last bit is, like Andrew said, tidying up your GenBank file. So we've said that you should clean up your product definitions. We've said that you need to add any type of notes that you find necessary. But Andrew has also shown that there are a lot of features that are additional to your GenBank file. Now, there's two trains of thought about this, whether any additional information is polluting your GenBank file or whether you are just reporting every bit of information that you can. I'm at this point, I'm not gonna take that much of a stance as long as you don't add the additional information that you have in your product or in your gene descriptions or in any of the feature qualifiers that matter. But if you were to want to remove things in a kind of systematic way, you can use the grep command with the v tag. And for example, if you want to remove all of the lines that start with inference, you can use this command, which we're gonna try out now. So I'm going to try this in one of my ones that don't matter that much. I'm going to do it in the original Proca form. So let's have another look at what that t4.gvk looked like. So we had a lot of inferences which said that the prediction was done with protocol. I'm just going to type what I found here. So it's grep v inference, and then, okay, my file name is only t4.gbk, and I'm going to name it something else. So grep v inference t4.gbk clean.gbk.
So if you remember, we said that grep looks for patterns line by line, but here we have the -v, that means invert match, and so only the lines without that will be printed.
Thank you, Andrea, because I'm not great with grep either. So we're gonna have another look at this file, and it looks like indeed I cannot see those inference lines anymore. Okay. So basically with this command, anything that you would put here, preferably which is at the beginning of your line, it will remove the whole line from your file. So you can use it for other types as well. One of the other things in our GenBank files, which we haven't discussed yet, is kind of the top bit. I'm just going to open it. I'm just going to open it again, where you can see, you know, your locus, your definition, so these are things that you can change within Eugene or within any type of text editor. I have matched some of the guidance from the INSDC website. So INSDC is an international sequence database consortium, which is a consortium that links NCBI, EMBL, and the database of Japan. So whatever database you submit to, they're all in a consortium, so they exchange data daily. So if you submit it to EMBL, the next day your data should be available from NCBI GenBank and vice versa. So they have some of the guidance that they have together on what is proper information to use. There's also very, very soon a paper going to come out by Andrew, Dan, and myself, and Igor from NCBI on, you know, some of the good practices on what information to include. Andrew has already mentioned that you should, you know, have a proper phage name. Don't add too much information in your definition line. Accession information gets added automatically after submission. So these are things that you cannot add. When you do a submission, there's stuff like papers, and so the publication in which your genome is first described that you can add, and that will also come all in this front bit. For this first bit, the features and location qualifiers source, which we call source modifiers sometimes, it is good practice to add as much information that you can for other people to use, you know, your genome with the specific metadata. So if you know the host, obviously you know the host because you've isolated the phage. You know the country in which it was located, all these type of things you can add here, and that's also information that is available on the INSDC website. For example, this actually gives you a list of the kind of qualifiers that you can use, and there's a lot. And there's a lot of it that's also not relevant because this is the same set of information that you give whether you have, you know, a human genome section or whether it's a phage. So, for example, what do we want here? Collected by is something that you may want to add. So you can add the actual person of who identified the phage, isolated the phage. The date, which is interesting, very interesting to have, because then we can follow up, for example, for T4, the different years in which it is sequenced and analyzed. There's basically a wealth of information that you can add. Country. My personal philosophy is the more the better. And once you have done everything, added all of this source modifiers, had added all of the information, removed all of the unwanted information, have all your product names, have everything perfect, then you can start submitting. And that is a whole nother can of worms to get the correct submission system. And we'll have to hopefully be able to expand our stuff here after we try it out again for ourselves, because it seems to change every other week. So I'm not going to give you any guidelines at the moment, because what I tell you might already be out of date. And I think we are coming to a close now.